Hi, I'm Lloyd Pye. Since February of 1999, I've worked with doctors, geneticists, and other scientific experts attempting to solve one of the most profound and compelling mysteries in the world today. That mystery is known as the Starchild skull, an actual bone skull that at first glance appears to be human-like. Nonetheless, 13 years of scientific testing have made clear it is not human, in whole or in part. Yes, you heard that correctly. It is not human in whole or in part. This incredible history-making relic was discovered in 1930 in a mine tunnel in the Copper Canyon region of northwest Mexico. It was found with a normal human female skull. Carbon-14 testing has revealed that both of the skulls died about 900 years ago. Every aspect of these two skulls shows stark physical differences that have been confirmed by multiple experts. Those differences will be thoroughly discussed in a separate video. However, this video will focus entirely on recent analyses of the Starchild skull's DNA using state-of-the-art equipment and techniques. DNA analysis boils down to only one term you need to know, which is base pairs, the steps on the twisted ladder spirals of DNA. Differences among and between base pairs determine genetic variation. In nearly all human cells, the nucleus contains the entire genetic package provided by both parents. This amounts to more than 3 billion base pairs, those steps in the spiral ladder of DNA. Floating outside the nucleus in the cell cytoplasm are many copies of mitochondria, which contain a separate small package of DNA that comes only from one parent, the mother. The first recovery of Starchild nuclear DNA produced several dozen fragments containing hundreds to a few thousand base pairs. Half of those fragments had base pair matches within the human genome, but that doesn't mean that the Starchild skull is human. Many animal genomes contain long stretches of DNA exactly like a human's. Chimps have 97% the same, gorillas 95%, rats 70%, and mice 65%. Where we struck gold was in the other set of the Starchild's nuclear DNA fragments, which did not match any known species. In and of itself, this did not prove anything, but it was strongly indicative that a large portion of the Starchild's nuclear DNA is highly unusual. Our next milestone was recovering 4 fragments of the Starchild's mitochondrial DNA. Remember, mitochondria float outside the cell's nucleus and are the power plants inherited from females only. Because mitochondrial DNA is so crucial to proper cell functioning, it very rarely changes. In the 200,000 years of human existence, our mitochondrial DNA has accumulated a maximum of 120 base pair changes. Neanderthals have around 200, the newly discovered prehuman Denisova has 385, and chimps quadruple that to 1,500. Altogether, the Starchild's 4 mitochondrial fragments have 93 differences that extrapolate to an astonishing total of 930 in the entire mitochondrial genome. Naturally, such a controversial result requires much additional testing for confirmation, but it is not unreasonable to assume the Starchild's mitochondrial genome will contain between 800 and 1,000 differences. Such a large number of them rules out a close genetic relationship between humans, Neanderthals, or Denisovans, and it provides yet another powerful indication that the Starchild is not human. In addition to the incredible results found in the Starchild's mitochondrial DNA, recently a fragment 211 base pairs long was recovered from the Starchild's nuclear genome, and it partially matched the mammalian FOXP2 gene. In normal humans, the FOXP2 gene contains 2,594 base pairs, and in each of us, the entire gene is absolutely identical. Because of this consistency, it is an ideal gene to compare between different species, including the Starchild skull. Here in blue, we see the 211 base pairs of human FOXP2 compared to the Starchild's fragment in black, along with several other species. The key things to notice are the red letters among the Starchild's fragment, each of which denotes a base pair difference. There are 56. Remember, normal humans have no differences in their entire FOXP2 gene, and certainly none within the 211 base pair fragment found in the Starchild. However, rhesus monkeys do have 2 such differences: mice 20, dogs 27, elephants 21, opossums 21, frogs 26. And if you extrapolate the Starchild's towering 56, out to 100%, the projection is a whopping 700 differences in its entire FOXP2 gene. As with the mitochondrial fragments, this astounding FOXP2 result is tentative. It must be confirmed with several subsequent runs in a DNA lab using next-generation sequencing equipment. However, with the Starchild starting in the range of 700, we can have that to 350, have it again to 175, and still be certain it will wildly surpass the literal handful of differences needed to completely and permanently separate the Starchild from humans. What on earth is the Starchild skull? Is it even from Earth? We certainly have powerful preliminary DNA evidence that strongly suggests it is something other than human, and it could be genetically remote enough from us to qualify it as an alien. But preliminary results are not the same as ironclad proof. So how do we secure that? Surprisingly, we can start here with the story of how humans came to fly. In 1901, Orville and Wilbur Wright brought their self-designed glider to Kill Devil Hills, North Carolina, to begin figuring out how to glide safely by controlling their craft in flight against the unpredictability of winds. By the end of 1901, the Wright brothers had learned that sustained flight had to be controlled in 3 axes of motion: pitch, roll, and yaw. This was their great discovery, which laid the technical groundwork for their ultimate success. But first, they had to negotiate 1902. In 1901 and 1902, the Wright brothers utilized a homemade wind tunnel to master their control of pitch, roll, and yaw. In 1902, they announced that they had solved the control in-flight problems, assuming something so revolutionary would find eager financial backers. Incredibly, that didn't happen. Why? Despite their success gliding at Kill Devil Hills, they were not recognized as experts among the world's first would-be flyers. Ultimately, the Wrights had to collect the money they needed from family and friends in their hometown of Dayton, Ohio, where they built the aircraft and the engine that flew them into the history books on December 17, 1903. And this is what we are trying to accomplish with the Starchild skull. Today, in mid-2012, we are exactly where the Wright brothers were 110 years ago. Like the Wrights, we took the Starchild's DNA to our version of Kill Devil Hills, a laboratory that figured out its genetic equivalent of pitch, roll, and yaw. Also like the Wrights, we have to secure the funding required to conclude our project by recovering and sequencing the entire 3 billion plus base pairs of the Starchild's genome, while filming the process for posterity and to confirm its validity. Now, imagine it's 1902 and somehow you know the Wright brothers are poised to do what no human has ever done before. They need help to make it happen, so they ask you to add a small bit of push to their problem. If you could have helped them then, how would you feel about that now? Well, we at the Starchild Project do need your help to spread this video as widely as possible, to reach the special people out there with the means and the will to join us taking the next giant step in human history. Unfortunately, we need considerably more money than the Wright brothers, but like them, we must find a way to prevail. Without them flying all of us into the world we know today, we could not have gone to the moon to take the first steps toward our inevitable future in space. Likewise, to transform us from isolated beings adrift in the vastness of the universe, we must first establish that a community of others definitely exists. That is what the Starchild skull can do for humankind today and forever into the future.